opinion, it doesn't matter if you went to the trouble to streak a sample on a plate or an isolate on a plate, or if you have a metagenome sample, you want to know what's in there. And for me, the first step is taxonomic classification. Computer scientists probably like to do a fast QC report to look at the read quality, but generally I start here. Now, what we're going to examine through the course of this analysis that we're going to be demonstrating is data from this particular study that was published by Street et al. And it's a really interesting study. They had 113 patients who had had an orthotic replacement, like a hip or a knee joint or maybe a shoulder joint. And then they got an infection. And generally what happens when you get an infection with these type of patients is they have to go in and remove the replacement and clear the infection before put it back in, because around where the joint is, there's poor immunity. So generally it's a big deal. You don't want to get one of these infections. So these 113 patients got the infections, and what they did was they removed the joints, and then they got sonication fluids that they used on the prosthetic joints and other metalwork to capture those samples there. And they sequenced them, and they put the read files in the sequence read archive. And so we're going to start with those samples. We're going to use the BV-BRC Taxonomic Classification Service, which uses several different bioinformatic algorithms, which we'll talk about. This service you can use for WGS, which stands for whole genome sequencing, or 16S sequencing, and it only uses sequencing reads. It utilizes several databases, which we'll talk about, and it gives you job results that include tables and plots, publishable figures, and also alpha and beta diversity metrics. So it's pretty useful. It's all based on, pretty strongly, there are some differences, but mostly it's based on this paper by Liu et al. That comes out of the Salzburg lab. They're a really powerful bioinformatics team, and they created this pipeline that BV-BRC has adjusted slightly. But these guys deserve the credit, so be sure to cite them and also cite BV-BRC, because we're providing you with the pipeline for your use. So like I said before, it involves different databases. If you're using whole genome sequencing, you can select to use either the Kraken-2 database or the BV-BRC database. And if you're using the 16S ribosomal RNA study, you can select between Silva and Green Genes. Now, this is what the pipeline looks like, and you'll notice that there are strong similarities if you start with whole genome sequencing or with 16S sequencing. There are just a few things that are different. Under whole genome sequencing, you can choose to do species identification, the pipeline's outlined in gray, or microbiome, the pipeline is outlined in blue. Both of them, it doesn't matter if it's whole genome sequencing or 16S, it starts with reads. Either paired like Illumina, single like PacBio or Oxford Nanopore, or you can load the run numbers from the sequence read archive, which is what we'll be doing today. The first step, for those of you who are computer scientists, it gives you a fast QC report right away. So actually you don't need to use FastQ utilities, but we're going to do that anyhow in other parts of the exercise. If you're doing whole genome sequencing, at this point, you can choose to align it to the host genome and remove the host reads and then separate out the non-host reads. If you're doing 16S, it will trim the reads, but it doesn't matter if you have trimmed reads or non-host reads, it goes into Kraken-2, and that's the powerhouse of this pipeline. And Kraken-2 gives you a number of things. A Sankey plot, which is pretty awesome, a multi-sample comparison table, a chronoplot, which I also like a lot, multi-QC reports. Now, if you are doing species identification, it will at this point generate a summary table. If you're doing microbiome or RNA-seq, you also get an abundance estimation that builds into the multi-QC report, but it also provides you with alpha and beta diversity statistics. So this is what it looks like. The landing page for the job at BV-BRC with your input file, the selected libraries file, and the parameters where you make all your decisions. And also at the top, it gives you links to find help, which we'll discuss later too. Don't panic. We're going to step through all of this so you can see exactly how to do it. And when it's filled out correctly, this submit button turns blue and you can submit your job. So when the job returns, some of the things that are returned with every one of these jobs are the Sankey plots, the Corona plots, both of which are a really great way to quickly visualize the immensity of the data that are in these samples and to bring your eyes right away to the things that you probably need to pay attention to. It also provides you with diversity statistics, and most importantly, it tells you what the diversity statistics mean, because I don't know about you, but I'm no longer a grad student or an undergrad. So if you ask me about Shannon diversity, I'm going to go, but if it's written there, I can tell you what it is. And also remember at the top of the landing page for the job, there's help for you. If you get lost, clicking on the information icon opens the user guide. Clicking on this thing that looks like a bunch of library books, one of which is falling over, takes you to the tutorial that gives you the step-by-step. And this little icon takes you directly to the YouTube page where you can watch a video showing you how to do everything. So we're going to step through this in the next video. We're going to submit a job, and then we're going to start analyzing it and go deep into this service. Thanks a lot. Bye.